We've already done the bacteria and the archaea, and we're going to now go through the rest of the eukaryotes, except for the plants, animals, and fungi. And then in the next lecture, we're going to talk about primarily one aspect of the diversity of eukaryotes, which relates to the organelles, the chloroplast, and the mitochondria. We'll talk about a few other related issues, but primarily the focus will be on these endosymbioses. We're going to have a quiz on Wednesday. I sent around an email about this. It's going to focus on the major groups of organisms. That's chapter 26.4 and chapter 27.5. The key here is to learn the major groups and some of the key features of the groups. And we're going to talk a lot about the eukaryotic ones today. The midterm will be Monday, in the second section on Monday. It'll be a mix of, it's very similar in structure to the practice midterm. I handed out a little shorter because the midterm is going to count for 75 points. That previous one counted for 100 because we didn't do the in-class quizzes before, so it'll be a little bit shorter. So hopefully hopefully no one will be pressed for time on the test. A mix of true, false, multiple choice, and sort of some either interpretation questions or short answer questions. And it's going to cover lectures 1 through 13, so up until the lecture on Friday. And we're going to schedule a review session. I'm trying to work on the scheduling of that, where you can come in and ask me anything you want, and try and just go over topics that you found confusing, etc. Yeah. Yeah, thank you. For the quiz, but not for the midterm, you are allowed to bring in a page, just one-sided, single page of notes, and write down anything you want. The goal here is to have you read and think about and then write down whatever you want about the major groups of organisms. Thank you for reminding me. Okay, so we're just going to continue on our tour here, and I'm going to highlight, in essence, some of the features or a feature or some key salient point about many of the different groups of eukaryotes that are out there. So, sort of as a reminder here, starting next week, we will start to cover plant diversity and evolution. It'll be Professor Keene, and then we'll cover fungal diversity and evolution. That will be me. And then the last 3 weeks of the course will be on animal diversity. And what I'm going to do today is talk about the rest of the eukaryotes. This is the tree from the book on the phylogenetic diversity of eukaryotes. I started on this right at the end of the last lecture. So the main points you should realize here are that there are 5 major groupings, which we will go through in detail today. The relationships among those groups, that is how excavates relate to chromalveolates, for example, is highly ambiguous. So this is shown as a polytomy. The branches, connecting and leading up to those 5 major groups, we do not really know what the order of those branches are. But the taxa that are assigned to the 5 major groups, that we're pretty confident in. So those are, all together in each of these groups. So you can consider that each of these groups represents a monophyletic lineage within eukaryotes. Again, even though we don't know how they relate to each other, each of them represents a major monophyletic lineage within eukaryotes. So one of the key things to pay attention to when you're reading the book and when you're listening in lecture, and we will talk about this in detail in the next lecture, is the presence or absence of organelles within the cells. So we'll talk a little bit about the evolution of the mitochondrion, which is present in many but not all eukaryotic lineages. And we'll also talk about the presence and evolution and function of the chloroplasts, which are present in some, and definitely not most, of the eukaryotic lineages, but it's present in quite a few of the lineages. So we're now going to go through the groups. I think I just barely started in on this at the end of the last lecture, so I'm just going to do it again. So the first of the 5 major lineages of eukaryotes are called the chrome alveolates. For most of these groups, both the 5 major lineages and some of the subgroups within them, you probably have not heard of these individual groups before or are not familiar with the names, but in many of the cases, you will actually know something about either the effects of some of the organisms or the individual organisms within the group. And that's what I'm going to try and highlight today, is either major sort of ecological or medical effects of individual members or taxa you may have heard of. So the chrome alveolates are divided into 7 major subgroups, and we're going to go through each of these here. Those subgroups are split into these 2 lineages, the alveolates and the stramenopiles. The book has a table that goes through each of the major eukaryotic lineages and some descriptive feature of that group. Many of these descriptive features are synapomorphies. We'll come back to that in a little bit. And also gives examples of some of the taxa in that lineage, frequently organisms that they talk about somewhere else in the book or that are, things you might have heard of. So, clicker question. Get you back into the groove on phylogenetic terminology. Again, for clicker questions, but not for quizzes, you are allowed to converse with your neighbors. So you're welcome to talk to people. And I'll give you until about a minute for this. What is a synapomorphy? Not supposed to use your notes for these clicker questions. You can talk to your friends, but don't use your notes. Please put... All right, I will give you -2 seconds. Come on. All right, everyone ready? I'm going to stop. All right. All right, most people answered B, a shared derived trait. That is the correct answer. Reasonable number of people answered A, a trait shared by monophyletic groups. Does anybody know why that's not correct? Yeah. So a trait shared by a monophyletic group or by multiple monophyletic groups could be an ancestral trait for that group, but presence of ribosomes is shared by animals. That doesn't mean it's a synapomorphy, right?
And also it's not derived. Like synapomorphy has to be derived, different from ancestral trait.
Yeah, I mean a synapomorphy, the formal definition is a shared derived trait. And for monophyletic groups, you can have things that are shared by all members of a monophyletic group, again, like the presence of ribosomes in mammals or in animals, but that's not a derived trait, that's something that's shared by all monophyletic groups. So the key for a synapomorphy is really they're distinguishing a particular monophyletic group from other organisms because that feature changed on the branch leading up to the common ancestor of that particular group. Yeah, a question somewhere? Okay. So the reason I wanted to get into that definition is in the book and in lecture today, we're going to highlight some of the synapomorphies for some of these groups. And when in the book or in the text or in lecture, if we don't explicitly say something as a synapomorphy, it might be that there's a feature shared by a group, but it might also be present in some sister taxa to that group. So it may not necessarily be a synapomorphy. It may be worth mentioning because many of the members of the group may have that feature. But usually we'll try and say that something is a synapomorphy. It's explicitly a shared derived trait for that group. So for example, alveolates have this interesting synapomorphy that distinguishes the clade, which is the presence of these little air sacs, basically, these little alveoli beneath the surface of the plasma membrane. And there are many of these. We're not going to go into a lot of them. I'm just giving you this as an example. That's where the name alveolates comes from, is the presence of this alveoli. So we're going to go through the different groups again within the chromalveolates, and I've tried to highlight up on the top of the slide. This is the chromalveolates in the lineage, the major clade, the alveolates, and now we're going to focus in on the apicomplexans. So the apicomplexans, again, the name may mean very little to most people, but many of the taxa in this group are familiar. So the causative agent of malaria, Plasmodium falciparum, is in this group. So responsible for millions of deaths worldwide a year. So again, even if you don't know what apicomplexans are, you're probably familiar with malaria. Toxoplasmosis is caused by an organism in this group as well. So there are many pathogens in this group. In fact, all of the taxa that are well characterized in this group are parasites. There appear to be maybe one or maybe a couple of, so far, free-living organisms, but the vast majority are known as parasites. The name comes from this apical complex. So it's part of the cell in these single-celled parasites that is used to, in essence, dissolve away the cells of their hosts. This apical complex is used to enter host cells. And that's a shared feature across the apicomplexans. The apicomplexans are a good example of something that is seen across many, although not all, of the eukaryotic groups, including, as we will see, the plants, animals, and fungi. There's an alternation of life cycles in the apicomplexans where the sexual reproductive stage, where haploid gametes merge together to produce a diploid organism, that takes place in a specialized environment in this case, during the infection process in mosquitoes, actually, which are transmitting the malarial parasite. And so in many eukaryotes, right, we have this alternation between the haploid stage of the life cycle and the diploid stage of the life cycle. And the timing or the exact occurrence of the sexual reproductive stage varies across organisms. Most organisms spend a lot of their life doing somatic cell reproduction or single-cell cloning, where a single cell makes a copy of itself by mitosis in eukaryotes. And then there's a specialized time of the life when gametes are made and sexual reproduction occurs. But most of the time, cells reproduce by mitosis. So we're just going to move on now to another one of the alveolate groups, which are Dinoflagellates. Most of them are marine. That means they live in salty water environments. And they are important photoautotrophs for the planet, producing quite a large fraction of the total photosynthesis on the planet. The name comes from the presence of these two flagella in a very specialized structural arrangement. One of them is on what's called this equatorial groove, and one of them is along this longitudinal groove in the cell. And you can see this little video. They use those two different flagella in the different orientation to swim around and to move and to do all sorts of crazy, bizarre things in some cases. We will learn a little bit more about them because dinoflagellates are symbionts of coral, and they're responsible for much of the biology that goes on in coral. They live in association with the coral organisms. They're also responsible for red tides. So if you live in the coastal areas around the planet, you might have seen some of these red tides. They frequently lead to fish die-offs and other problems, and many of these red tides are caused by dinoflagellates that produce a toxin that then kill a lot of organisms. So you're probably familiar with a lot of the byproducts of dinoflagellates, even if you're not familiar with individual organisms. Third group in the uvulates are the ciliates. Should I shut the front lights here? At least dim them down. I don't know if that helps at all. But so the ciliates, as the name suggests, are characterized by the presence of these cilia. They're small, hair-like projections on the outside of the cell. Most of the members of this group are heterotrophic. In a way, you can view them as predators. They sort of swim around and eat bacteria and other small organisms in their environments. Really interesting feature of their biology is that all of them actually have two nuclei, rather than what you might imagine, the typical eukaryotic cell with one nucleus where the DNA is packaged. You can view ciliates as, in essence, a single-celled version of an animal body. One of the nuclei participates in sexual reproduction and serves sort of like the germ cell lines do in animals, reserved, protected, and saved for sexual reproduction. And the other nucleus is basically the equivalent of somatic cell, the body of animals, and it participates in expressing the phenotype of the cells. So this is inside a single-celled organism, is this sort of complicated diversification of roles for the different nuclei. You're probably familiar with some of the members of this group. So Paramecium is sort of the classic model single-celled organism used in a lot of biology laboratories. If you're familiar with how Paramecium works, most of the other ciliates work in some sort of similar way with these, pumping food inside of them and digesting the food in these vacuoles inside the cell. There's incredible diversity in form among ciliates. There's all sorts of wild, like this, Halloween pumpkin-like ciliate. There's all sorts of interesting forms that ciliates take, and they have lots of diverse roles in terms of what they do in different environments on the planet. So another group that the major name you probably are not particularly familiar with, but you will be familiar with some of the representatives of this group, are the Stramenopiles. And the Stramenopiles are made up of three main lineages: the brown algae, the diatoms, and the oomycetes. And you will be familiar either with organisms in each of these groups or with the effects of them. The Stramenopiles have a synapomorphy. Many of the synapomorphies for these different groups relate to structure of the cell and or how they arrange their flagella. Those are the ones that are known. And in this particular group, they actually have a dimorphism in the flagella. There are two flagella. One of them is longer than the other, and that one has these rows of tubular hairs on it. So here's an example of one of the Stramenopile lineages, the brown algae, which includes kelp. All of the representatives of this group that are known are multicellular, and some of them, again, like kelp, growing to be hundreds of feet in length. So this is important to realize. Multicellularity has evolved separately many times across eukaryotic evolution and also in bacterial evolution. You may be most familiar with multicellularity in plants and animals, maybe mushrooms and things like that, but there's multicellularity across the eukaryotic world. The exact mechanisms by which these organisms carry out their sort of transport processes and communication and development vary quite a bit across the eukaryotic world. The brown part of brown algae comes from this fucocyanin carotenoid, and these are almost exclusively marine, that is, again, saltwater environments. And we'll hear more about brown algae when we talk about evolution of chloroplasts in a little bit. Diatoms, you probably heard the name. quite a bit. You may not be familiar with too many of the individual organisms. They are unicellular most of the time, although some of them live together in sort of these colonial filaments. They have quite a diversity of colors that come from these carotenoid pigments. One of the most important things about them is their excellent fossil record. These are fossilized diatoms. Because they make these hard extracellular surfaces, they form spectacular fossils. And we know an enormous amount about their evolutionary history because of these fossils. They also, in the marine environment, they die and they fall down into the sediment. That's why we get lots of good fossils from them. They also then, after other sediment piles on top of them, degrade and turn into gas and oil. That's where a lot of gas and oil reserves come from degrading diatoms. In the oceans, they're estimated to be responsible for 20% of all carbon fixation on the planet. So very important ecologically. And we will also see more about their photosynthesis and chloroplast evolution in a little bit.
While these are protists, they form
the
some
third
of the
lineage
largest
within the
organisms on the
Stromatopiles,
planet,
you've probably not
the giant
heard of this
kelps
group
of the
really
marine
at all.
forest.
They're called the oomycetes. Many of the things that end in mycete are things that people thought were fungi because they form these structures that look a lot, these branching filaments like fungal structures that you will see in a couple of weeks. These are not fungi. They're not even remotely closely related to fungi. They're all pretty much sort of parasitic heterotrophs, living off of other organisms like growing on this dead bug. And reasons you might be interested in them include that the representatives of this group include the causative agent of the potato famine, the potato light blight, which wiped out potato crops multiple times separately throughout human history. It also includes the causative agent of the sudden oak death that's going around California in particular, as well as other parts of the West. So it's really important to understand what these things are. When they were misclassified as fungi, people tried to treat plants infected with them with antifungal drugs. They don't work particularly well when something is not a fungus. And so people have had to develop other strategies for dealing with these once it was discovered that they are not fungi and not even closely related to fungi.
This is a
So the last group within the chromalveolates is another group that
highly specialized
the name of the group may not be that familiar, haptophytes.
The cells are
It
linked
includes
by a tiny groove
organisms
system
like these
along their silica
coccolithophores
wall.
that can
Movement
form
is generated
huge
by
sort
an
of
active
single
system
species
coordinated in the
blooms
colony.
in the ocean. This is imaged by satellites. So this is probably hundreds of trillions of individual cells from this particular species. Those blooms can have major effects on the oceans because they block out all light and they use up lots of nutrients and oxygen and can kill basically everything in the environment. There's another really interesting organism in this group called Emiliana Huxley, which people are just learning about basically in the last 5 to 10 years and so. When people go out to study a lot of the marine sort of plankton in the oceans, one of the things they do is they filter the water. So if you're interested in diatoms or microbial eukaryotes, what you do is you filter out the big things like the fish and the sharks and the jellyfish, and then you let the water flow through and you filter the water so that you get small things, but things that are bigger than bacteria. And then you study those. Unfortunately, Emiliana Huxley is smaller than even some bacteria, and it was missed by these types of filtration efforts. Because the cells are so incredibly small, they went through most of the filters and were largely ignored. So we're going to move on to group number 2 here. The Plantae, which you will hear lots about in a week or so, because this includes the land plants. We will spend 2 weeks on in the class. The Plantae are divided up into 5 major lineages. All of them include photosynthetic organisms, as we will see in the next lecture later today. It is in this lineage that it is thought where the chloroplast symbiosis originally happened. Again, in the book, there's a table with key features of these organisms. If you see the glaucophytes, one of their key attributes is peptidoglycan in chloroplasts. So if you look at the chloroplasts inside these glaucophytes, they actually have a cell wall, which is unusual for chloroplasts. Most of the glaucophytes are freshwater, single-celled organisms. But again, many of them have peptidoglycan in their cell wall. So another clicker question. Why would I tell you that there is peptidoglycan in the cell wall here? What is peptidoglycan? Again, no notes, but you can talk to the people around you. I'll give you until about a minute on the clock here. All right. 5 more seconds. Hurry up. Put in an answer. Come on, come on. All right. The majority put A as their answer. Found in gram-negative and gram-positive bacteria. That is correct. So peptidoglycan is one of the chemical components of the cell wall of bacteria. The bacterial cell wall is thicker in gram-positive organisms and thinner in gram-negative, but they both have cell walls and they both have peptidoglycan in the cell walls. Archaea do not have peptidoglycan in the cell wall. So the reason that this is important for glaucophytes is that that presence of peptidoglycan in the cell wall is a good indication that the chloroplasts are derived from bacteria. Peptidoglycan is only found in bacteria. The chloroplasts look like cells. They look like they could be archaea or bacteria or some simple cell, but the presence of peptidoglycan is a really good indicator as to what their evolutionary history is, and we'll talk about that again in the second lecture later today. So the next group, if you watch the video, there's this really cool sporulation going on. The next group of plantae are the red algae. Most of these organisms are marine. Most of them are multicellular. It appears to be a separate origin of multicellularity from what we see in land plants and in some of the other algae. Many of them reproduce by this formation of spores that they then release into the marine environment. That's why I was showing some of the videos. Many of the spores are actually motile. They have, I think, flagella on them that allow them to sort of swim around in the water rather than just being moved around by diffusion.
Red algae are generally
chlorophytes are the sister group of this clade here, which we will talk about later in the class. The land plants and charophytes are, in essence, the main things we will talk about when we talk about plant evolution. So the chlorophytes are the sister group to that. They're incredibly diverse. There are something like 17, I think it's even more now, like 20,000 species that have been characterized, and they're found in marine, freshwater, terrestrial, all sorts of different environments. There are single-celled forms, there are multicellular forms, quite a lot of diversity here. They're all basically photoautotrophic. This whole clade has some synapomorphies that relate to use of particular chlorophyll compounds that we'll hear about later in the class. And again, we're not going to talk today about this group at all because those will be covered when we do the plant lectures.
This sequence shows the Volvox
All right, so
colonies
the
as
third
they swim.
major
They
lineage
move through synchronized
are the
Phylogenetic
excavates,
in which strong currents and is able to.
Again, the book has in the table a decent summary of the key features of these groups. There are 5 major lineages of excavates. These 2 up here are really important for studies of eukaryotic evolution for one reason in particular, and that's because they don't have mitochondria. So the diplomonads and parabasaliids are some of the organisms among eukaryotes that do not have mitochondria and have been the subject of a lot of debate about what they mean in terms of the evolutionary history of mitochondria. The Giardia causes a very unpleasant gastrointestinal ailments, is caused by a diplomonad. Trichomonas is another member of this group that also doesn't have mitochondria. And as we will see when we talk about mitochondrial evolution, the conclusion is that these organisms, their ancestor had mitochondria and they have subsequently lost their mitochondria. They're all unicellular in these groups, and most of them that are characterized at least, are parasites. Heterolobosians, which, again, another group you probably haven't heard of the group name, but you've probably heard of the grain-eating amoeba. And in lab, you're talking about a relative of one of these organisms. So there are members of this group that cause major human and animal diseases, so they've been reasonably well studied. Most of the other members of these groups are not very well characterized. Many of them have this amoeboid body form, and as you're talking about in lab, many of them can switch back and forth between different body forms. And generally, the morphology of a bacterium or an archaeon or a eukaryote is not a great indicator as to what its evolutionary history is, partly for this type of reason. So for many years, these organisms were misclassified where the flagellated form was called one thing and the amoeboid form was called another. And only with DNA analysis was it figured out that they were the same exact thing that just shifted shape. Fourth group within the excavates are the Euglenids. You may have heard of Euglena. It's a photosynthetic, single-celled organism. Many of the organisms in this group sort of swim around inside their aquatic environments. Some, but not all, members of this group are photosynthetic, and there's some really interesting aspects to the evolutionary history of their photosynthesis, which again we will see in the next lecture. Some members of this clade can switch between photoautotrophy and heterotrophy. So they are not obligately photoautotrophic. They can actually consume things from their environment as well. And then the last group within the excavates are the Kinetic Plastids. They are characterized by the presence of this specialized structure called the kinetoplast, which is inside these organisms' mitochondrion. In some texts, although I think they've corrected this in our book, and in some other places you will see these described as plastids rather than kinetoplasts. You might mistakenly think that they are chloroplasts because of that terminology, but this is actually a specialized structure inside the mitochondrion. It is not related directly to chloroplasts. This group is pretty well studied because it includes the causative agent of diseases like Chagas disease and African sleeping sickness and Leishmaniasis. One of the key features of this group is that they all have just a single mitochondrion. So in many human cells, for example, there are hundreds to thousands of copies of the mitochondrion. In these kinetoplastids, there's just one mitochondrion inside each cell. Yeah?
So trypanosomes are a subset of kinetoplastids, and trypanosomes definitely cause sleeping sickness. And they cause Leishmaniasis and Chagas too?
So trypanosomes are a subgroup within the kinetoplastids. They are very closely related to the Leishmania group that causes Leishmaniasis. So the trypanosomes cause Chagas disease and sleeping sickness. And all of the members of that group, they're transmitted by insect vectors. There are different insects for each one of them. So some of them are transmitted by mosquitoes. I think Chagas is transmitted by, I think, a beetle or a bug, but lots of the parasitic organisms in the eukaryotes go through a vector as part of their life cycle to be transmitted between hosts. And many of them can't survive on their own outside of a animal sort of environment, so that's one of the reasons they use vectors. And many of them are blood-borne, so they need a blood-feeding insect in order to be transmitted. Okay, so group number 4, you'll be happy to know it has only 3 major subgroups within this group. The Rhizaria.
This real-time
Rhizaria includes 3 major lineages. Again, probably have not heard a lot about the Cercozoans, the Foraminiferans, and the Radiolarians, but you will be familiar with many of their effects, at least of the Foraminiferans. So the Cercozoan, there's a characteristic feature, which is that they're generally unicellular and aquatic and have these long, thin pseudopods. The Cercozoans are very poorly understood. Some are aquatic, some live in soil. One subgroup of them has chloroplasts, which we'll talk about when we talk about chloroplast evolution. But generally, they've been basically very poorly studied and only really known largely due to molecular work that's come about in the last 10 or 15 years, that someone had characterized one of these and just sort of threw it off to the side. And then someone built an evolutionary tree and said, wow, this is a novel lineage. Let's learn more about it because it's distantly related from everything else out there on the planet. But there's just not a lot of biology known about these groups. The Foraminiferans, on the other hand, there's lots known about them, partly, like with the diatoms, due to their fossil record. So they secrete shells of calcium carbonate, and these shells fossilize really well. And the dead animals and their discarded shells are what makes up limestone. So that's where limestone comes from, the shells of these organisms, as well as many sandy beaches. The sand comes from the shells of these organisms. And again, they have an excellent fossil record. Many of them live as plankton in the marine environment, although some are actually on the bottom. And they have this really interesting biology, which is through these shells, they stick through these branched pseudopods, and they coat them in this like mucousy, sticky substance that allows them to capture plankton in their marine environment. So they sort of float around like flypaper or something to that effect and accumulate all sorts of food on their sticky pseudopods and then eat that. I think I briefly mentioned this one before in the class. This is a foraminiferan called a xenophyrophore that I have a little video of. So I do some deep sea research, and on a cruise that I was on, they sent down the Alvin submarine into the bottom of the ocean. I was not in the submarine at the time, but a graduate student from Woods Hole was there, and she had been working on a book about the diversity of eukaryotes in the deep sea, and she knew what this thing looked like. So they were cruising around in this submarine with these tiny little portholes, and the submarine pilot was just going to blast on past this thing and go looking for like sharks and fish and other big things. And she starts screaming out, no, you have to stop, because this is this weird foraminiferan the size of your fist. It's a single cell, many nuclei, but a single cell the size of your fist. They crawl around on the ground, leaving trails behind them. They're very, very strange organisms, very poorly understood. And she knew sort of immediately that this thing was interesting. So this is actually a photo taken from the cruise by I don't know who. And if you Google xenophyrophore, which is the name of this organism, this is the picture in Wikipedia for it. And we actually have this individual cell in the freezer in my laboratory. So we've been trying to study it from this sample that was taken in the deep sea. And so the third group of the Rhizaria are Radiolarians. Also, you may not be that familiar with them, but like the diatoms and the foraminiferans, they form these very hard skeletons. In this case, they're actually glassy endoskeletons, but they make really cool structures, and they form, in many cases, excellent fossils. So there's a reasonable amount known about their history because of these structures. And this is really important to realize. Many organisms on the planet might have an incredible diverse history throughout geological time. But if they don't form fossils, we may know very little about their past evolutionary history. We can characterize what they're doing now, but their past history, we can only study it if those organisms form good fossils. So with most bacteria and archaea, they've been around the planet for billions of years. They don't form good fossils, so we don't know a lot about what they were doing in the past. All right, so the last group of eukaryotes are the Eunuch Anse. Again, here's a description of them in the book. That table has lots of detail in it. The Eunuch Anse are characterized by the presence of a single flagellum. That's where the euna comes from. That is, if they have a flagellum. Some of them don't, but they never have two, like you saw with Dinoflagellates and with many of the other eukaryotic groups. They always just have one or zero. Fungi and animals are in the Eunuch Anse. We will talk about those more later in the class. Fungi and animals are in this subgroup called the Opisthokonts. And you will see from this tree that there's one lineage here, the Dinoflagellates, that is actually the sister group to animals. In essence, more closely related to animals than any other group on the planet. Dinoflagellates are quite interesting. There's a lot of studies of them as models for understanding the biology of animals. Many of them are colonial, form sort of little groups of cells. The name comes from what the cells look like. The cells look like certain type of cells seen in sponges. And you will hear more about this when Professor Keene talks about sponges later in the class. But there a lot of work done on this group to try and understand the early events in animal evolution by studying their sister lineage. The amoebazoans are the last of these groups that we're going to talk about. They're generally characterized by feeding by phagocytosis and living as sort of predators and scavengers, although there are a few parasites as well within the group.
This
One of the lineages is called the Lobosians.
Captures and swallows
Again,
food.
you may not be familiar with the
This
name of the
sequence is
group, but
shown at
includes
real
things
time.
like Entamoeba histolytica, which is a human pathogen, sometimes found in municipal water systems, for example. Some of them, like the foraminiferans, sort of secrete this mucousy glue-like structure. They sometimes use it to sort of collect a little house around them, made out of
indicate the
small particles of
food
dirt or
items
sand.
that are about to be ingested.
There are a reasonable number of pathogens in this group, again, like the Entamoeba histolytica, all living as single cells. I don't think any are known as multicellular or even colonial. There are two groups that are called slime molds. Watch this little video of it crawling across the surface and eating something. There are two groups called slime molds. In this tree, they are grouped together with the plasmodial slime molds and the cellular slime molds. It's not clear if they really are sister taxa. They are clearly different groups. So there's this one called the plasmodial slime molds. The term plasmodium basically means a single cell that has many nuclei. So what happens sometimes in eukaryotes to get a big cell like the xenophyophore and like some of these is a cell can make copies of the nucleus, make copies of the genome, and not split the cell in half. So you have many copies of the genome inside one single cell. And that's frequently referred to as a plasmodium. Many of them form these stalks and spores where they look sort of like some fungi that we will see later in the class. And many of them do this sort of crawling across surfaces. And then the last group are the cellular slime molds. Many of them have this life cycle where there's individual motile cells. This video is showing them over here that sort of swim around and ingest food by endocytosis. And they eat and eat and eat until they, I guess, aren't that hungry anymore and get together with their buddies and form a reproductive structure. And this is called a fruiting body or a fruiting structure. And then this is, leads to, in essence, reproduction of the individual colony here. So this is a simple form of multicellularity and is used as a model system to understand the evolution of multicellularity. This organism, Dictyostelium, is a model for many molecular and cellular studies of eukaryotes. And it's just going to keep on going until it starts to grow that stalked body. And there are a lot of these other eukaryotes that do form these sort of complicated reproductive structures. We're going to focus in on the reproductive structures of plants, animals, and fungi, but there's lots of other diversity out there across the eukaryotic world.
An amoeba moves across the field
again,
at 25
we
times
microbiologists
real speed.
like to think that movies
This
are based
cell
upon
has
what we
eaten
work
numerous
on.
green algae, now contained in
Probably not true.
digestive
Someone could
vacuoles.
easily have just invented
Movement
the
is
blob,
generated
but
from
we're gonna say they're slime
cytoplasmic
molds.
streaming by
So the last thing I want to talk about in the last few minutes is evolution of one group that we haven't really talked about a lot. And these are the viruses. It has come up in a couple of questions, and I've mentioned them peripherally, and there's some in chapter 26 in particular about them. So if you look at viruses that are out there, viruses are basically pieces of nucleic acid, either DNA or RNA, surrounded by a protein coat that are parasitic and infectious. They make use of cellular organisms, that is bacteria, archaea, or eukaryotes, to reproduce and then spread to other cells.
This
There's
acellular
incredible
slime mold,
diversity
Physerum,
among
is
the
a single
viruses.
cell enclosed
Again, with
by
all
the
sorts
cell
of different
membrane.
protein
It grows
coats,
outwards
different
by
types
rhythmic
of
pulsations
DNA
of
or
cytoplasm.
RNA that they
These
use as their
pulses are
genome. Some of them
driven
have
by
multiple
shuttle that is
chromosomes,
back
like
and
flu.
forth streaming in the
Some of them
cytoplasm,
don't.
driven
Many
by
of
the
them have
actin
very interesting
cytoskeleton.
mechanisms by which
This
they
sequence takes place
hurdle
over
themselves or
2.25
project
hours.
their DNA into cells of their targets. There's just incredible diversity across the viral
cellular
world.
slime mold, Dictyostelium.
Lots of diverse phenotypes. Many of them are, cause diseases that we know about. Other ones, they infect things but have no sort of obvious disease, although they might have effects on their host. And what I want to end with today is talking very briefly about viral evolution. So the fact that we haven't included viruses on the tree of life does not mean that we don't think viruses evolve. We know they evolve. They change over time, they respond to natural selection, they evolve in the same general ways as other organisms, But we don't consider them alive, and really we don't know where to put them on the tree of life. And one of the reasons for that is there's still a debate about where viruses come from. And I've listed here the three main theories for the origin of viruses. The first is that viruses are relics from an ancient pre-cellular world. That is, early in evolution, when even before the existence of the most recent common ancestor of bacteria, archaea, and eukaryotes, maybe there were pieces of DNA that were evolving, and some of them became parasitic, taking advantage of other pieces of DNA or RNA, and as primitive cells evolved, those parasites stayed as parasites and never became cells. And all along the way, as cells evolved, these parasites stayed as a separate, distinct lineage. You could call it the fourth domain, or something to that effect in the pre-cellular world. A second possibility is that a part of a cellular organism, in essence, escaped and became its own entity. Inside all organisms on the planet are mobile pieces of DNA called transposable elements that move around inside our genomes. Many of these transposable elements function in ways very similar to viruses, except they're not infectious. All you have to do is add a little protein coat around them, and they become viruses. And so many people think that this is a possible explanation for the origin of some viruses. And then a third possibility is that you had a parasitic cell like a bacteria that had already thrown away its cell wall, that was becoming an obligate intracellular parasite, like chlamydias, and it just threw away even more of itself, and eventually lost its membrane and became a virus. It turns out that all three of these are probably correct. Viruses are almost certainly polyphyletic. Different viruses have different histories. Some of them probably are relics from the precellular world. Some of them are probably escaped portions of cellular organisms. And some of them are probably extremely derived parasites. So all three of these appear to be possible. The problem is we still don't know for most viruses which they fit. I will see you later today.